Hi all. Welcome to the inaugural session of the QCMS online seminar series. The full form of QCMS is QSAR, Cheminformatics and Modeling Society. The current form of QSAR Society, that is QCMS, was founded by Professor A. Cavalli, University of Bologna, Professor G. Costantino, University of Parma, Professor G. Crucieri, University of Perugia. And the current board members are Professor Giovanni Bottegodi, University of Urbino, Professor Andrea Cavalli, University of Bologna, and Professor Rebecca Wade from Heidelberg University. The QCMS Society has planned to organize an online seminar series on an approximately quarterly basis to interact more with the Cheminformatics and QSAR community. With the help of my fellow members, Tudor and Abdur Rahman of the QCMS webinar committee, I'm very pleased to conduct the first session of the series. Today we have two speakers. Our first speaker is Professor Alexander Tropsha. He is K.H. Lee Distinguished Professor at the UNC-Eshelman School of Pharmacy and Chief Domain Scientist for Molecular Informatics at the Redesa Computing Institute, UNC-Chapel. Professor Tropsha obtained his PhD in chemical enzymology in 1986 from Moscow State University, Russia. His main research interests are in the area of data science with application of drug discovery, chemical safety prediction science. He has authored or co-authored approximately 350 peer-reviewed research papers, reviews, and book chapters. He is an elected fellow of American Institute of Medical and Biological Engineering and a consultant to several technology and drug discovery companies. Today, he will speak on reminiscing about the future of Cheminformatics. Professor Tropsha, the floor is yours. You can share your screen.
Thank you. Let me try that. And get to the right place. Okay, can you see my screen and can you hear me?
Yes.
Okay, so let me get started. Please let me know, Kunal, if there are any issues with transmission, voice or visual. But thank you very much, and I'm very pleased to, I guess we're inaugurating this series of online talks, so I'm very pleased to be invited and to speak today. And because it's one of the first presentations in the series, I have decided to be to give a sort of an overarching lecture instead of focusing on any particular topic in cheminformatics and talk about, from a somewhat sarcastic but optimistic viewpoint. As you will see, I will go through some exciting events as well as criticism of methods and applications that have emerged in the field in the last few years. And I think it would be fair to say that the current state of our field can be seen as some sort of Olympics. It's symptomatic that this lecture is happening at the same time as Winter Olympics. We are trying to be faster and do more and do it cheaply. So why? And I want to explain the title of the lecture. I think it's very important to reminisce about the future from the psychological viewpoint. This is the definition of reminiscence, which is recalling episodic memories from one person's past. And I'll try to recall some memories from the history of the field, but this process often triggers thoughts of the future. And we have an opportunity to live in present, which is lasting, in our case, about 40 minutes until I'm done or interrupted. But it allows us to look into the infinite past and infinite future and perhaps together decide what is important for us to work on and what is not important for us to work on as we go through our research projects. So reminiscing a little bit and go into not necessarily the beginning of the field of cheminformatics, but an important event in the history of the field where the Journal of Cheminformatics just started. And what you see on the screen is a summary of challenges that have been defined in the editorial in the beginning of the journal that I hope, or I think many people online have contributed to, read it, or wrote papers. And Karina, who will give the next lecture, recently became the associate editor of this journal. So hopefully she'll talk about this journal a little bit as well. But that's the challenges that have been defined in the beginning. And number one challenge was our common stalled drug discovery, and that was more than 15 years ago. Now, you could be thinking big, but in reality, when we work on cheminformatics and kind of about the same years as the journal had started, or within the same time frame, our group has been actively working on what we consider basics of the field, including best practices for model development, validation, exploitation. Again, another paper on validation, paper that emphasized the importance of chemical structure and chemogenomic biological data curation. And all of that was, we thought was foundational for the field to achieve success in the experimental drug discovery work. Meanwhile, as you all probably know, the drug discovery field continues to attract enormous interest and attention and investment on the part of biotech and pharmaceutical firms. And yet, as was noticed in a famous Nature Reviews Drug Discovery paper, the evolution of the field has been likened more to what was called an Irum's law as opposed to Moore's law, given the sharp rise in the cost of pharmaceutical R&D, while the drug approvals continued to be low. And so that the trend that has been picked, with some hope, I guess, emerging in around 2010, that the trend had stabilized but has not really gone up, was that we continue to invest a lot and we continue to discover a relatively small number of drugs. I'll talk in a minute about how many drugs we currently have. And this particular slide or picture is taken from the FDA CEDAR report just published in January of this year. And we could see that the number of approvals continued to fluctuate around 45, 50. Pretty much across the last decade. And so there's clearly a problem with the number of drugs and the number of unaddressed problems that continues to be a central problem in the field. And since some time ago, I started asking these questions when I give lectures about the total number of approved drugs. And it's really not very well known. People give numbers anywhere from 1,000 to 15,000. But we recently, and that's January of this year, we published a paper where we used large language models to analyze textual data from various documents, federal documents, FDA in the US, EMEA in Europe, and then national agencies similar to FDA in Japan, Russia, and India, to count how many drugs are registered as approved drugs, co-formulated drug combinations. We also counted through a separate effort the number of registered diseases. Diseases have been redefined, as you probably know, and then cancers and tumors. And what emerges is that the total number of approved drugs, legitimately approved drugs, is less than 4,000. While about 5,000 diseases continue to have no treatment, with many diseases only allowing for partial treatment by existing drugs. And so that continues to create the problem that was formulated in the Journal of Cheminformatics in the beginning of the journal, that we continue to observe stalled drug discovery. And we all, I think, and should be thinking as to how to make this process go faster. And I will offer a thought as to what our community can do to accelerate this process, as well as to define acceleration toward the end of my talk. But progressing from 2009 and the years when we and others have been defining what is important for the field and how important it is to validate models, curate data, etc., progressing to 2024, 2023, where Pat Walters started publishing a series of blogs and papers outlining the consistent problem in the fields. And the problems were not to overcome high-level problems, to overcome stalled drug discovery. The problems that Pat had been consistently emphasizing in his blogs and talks and presentations, and I do encourage you to listen to him every time you have an opportunity. They are, if you will, pedestrian main themes for the field. We need valid structures, consistent measurements, realistic range of data, data curation, model validation metrics. All the same problems that we and others have extensively published on 15 years or 20 years ago continue to plague the field. And the situation is probably even more acute as we have more data and focus on more and more methodological development, something else that I will plug into my presentation. And so what this tells us is that we need to constantly revise the approaches that we use and problems that we face and want to solve in front of us. And what we need is, and what seems to be emerging, is new, big, high-quality data, maybe new methods. That's a controversial thought as to whether we need a flurry of new methods and new success metrics. And this is what I'm going to talk about, reviewing what we have currently in the field, which seems to be big data as we look at this, and we'll look at the publicly accessible resources for building models with about 3 million compounds assembled in Campbell, which is a relatively highly curated database of chemical bioactivity, with PubChem, which is less curated but much bigger. The current count is it's a little bit outdated, about 130 million molecules and almost 300 million bioactivities. So it's still large database that we could explore for the purposes of drug discovery. And more we are developing tools with what I discussed as our MEDIC database of approved drugs, large language models, in order to extract deeper information from texts and publications and tables and figures published in the literature. And we could analyze all this data, on one hand, build models of this data, and then use this data to mine what is really emerging to be a huge, ultra large databases of purchasable, fully enumerated chemical structures. So there's a big technical and functional challenges to how to mine this information using various machine learning and knowledge mining and virtual screening tools to identify new molecules that are purchasable, as well as use generative chemical design to come up with more predicted actives informed by the models built with what appears to be a large database of measured chemical bioactivities. So these are the overarching challenges in the field, and this is the introductory part of my lecture to outline the type of data that we seem to have available and the type of problems that I annotated here as, in my opinion, addressable in the big data age of cheminformatics research. And I will go through examples of studies that were done in our lab, predominantly talking about what I consider important areas of research, which is rigor and accuracy in data retrieval and preparation, where any machine learning exercise starts. And I cannot emphasize enough the importance of rigor and accuracy in processing primary data accessible to us for building models. And what emerges as a result of the huge growth of purchasable compound collections, the importance of rapid and accurate chemical similarity search, similar developments in structure-based drug discovery, where the same libraries need to be processed, but using the information about protein structure and calculating rapidly docking scores in virtual screening exercises. And I will also offer an opinion on how we should change the metrics of assessing the accuracy of machine learning models, given the huge imbalance between the training set size and the virtual screening library size. And I'll talk about positive predicted value of special type in the last portion of my talk. I will not talk about, for the lack of time, of what really is emerging as a very important area in the field, which is accurate and rapid property predictors, because virtual screening hits typically come when you screen multi-billion compound libraries, come in big scores, and we need to filter them for physical, chemical, and other drug-like properties. And so the tools that have been developed in the field to screen those compounds and identify more plausible candidates for further optimization are very important, to the point that JMed Chem published an editorial a couple of years, less than 2 years ago, suggesting that if we do this well, somebody will get a Nobel Prize. So here is a suggestion for younger people in the audience as to what to focus on if you want, if you believe this prediction. So let me start with rigor and a little project that we've done just to illustrate how important it still is to work on what we and had in his most recent blogs highlighted as a critical part of the field, which is data curation. And this is a small project that we published a couple of years ago with a very simple task. It was sort of in the midst of pandemic, and the task was to extract and annotate all chemicals that have been tested in direct antiviral assays organized in Campbell. And our initial thought, knowing that this database is well maintained, was that that's going to be a relatively simple, straightforward exercise, maybe taken a day, because the assays are organized, and a simple query for assays established and published for direct antiviral compound discovery is going to be a simple exercise, and then we would analyze this and maybe identify molecules worth pursuing further that have multi-antiviral activity. It turned out, however, that the exercise was not as easy as we thought it would be, because as we started analyzing the data, and of course, when you analyze assay data, there are important attributes of the assay that may or may not be reported. When we build a QSAR model, all we want to know, without diving deep into how the assay was done, is a number that characterizes compound bioactivity and the chemical structure, and that seems to be enough to start building model. But it's really important to be very mindful of how the assay was conducted, and when we integrate data, especially from different labs, how concordant the measurements are and whether or not we could truly integrate the data. And what we found, looking into the specific fields associated with every assay, is that as integrated as the data is, and as careful as Campbell team had put things together, there were a lot of inconsistency in the characteristics of compound bioactivities, with the best example shown on the right, where pretty much every important piece of information as to how the assay was done was included. But many assays would just have a simple description, antiviral activity against SARS-CoV-2, with a lot of data being in the middle, where you really would not have enough information to analyze and put data together. And so what we found through a lot of efforts and a lot of very tedious manual work of a graduate student working on this particular project is that a lot of results were missing information that we have to mine either from primary publications, or the information was misaligned and was not accessible in the columns in Campbell that advertised that this information is available. And so through this lone efforts. The paper is published, but this is just an example of how much time it took for a well-trained graduate student to extract and organize and consolidate the information in the proper format before any data analysis was established. To the point that if we used a very simple query of the Campbell database, we would have lost almost 100% of all collected data. And so we had to be very nosy and very specific in our prompts and the analysis of the data to eventually arrive at the database that we called SMAC, Small Molecule Antiviral Compound Collection, and the effort was published in Antiviral Research a couple of years ago, with a total database that contained, as you could see, thousands of drug assay pairs, 12,000 compounds, and well-curated efforts. And this database is publicly available from our lab. And I wanted to start with this little story just to emphasize that as much as we want to focus on new algorithms for model development, what really the core of cheminformatics is working with the primary data and preparing data for subsequent execution, building models and execution. Going on to rigor of modeling, something else that we and others have emphasized. As we all know, the excitement in the field in the past 10 years has been associated with the emergence of deep learning approaches. And that's the next topic I'd like to talk to in perspective in the context of reminiscing about the future of machine learning as applied in cheminformatics. A group of us published this review in Nature Reviews Drug Discovery less than 2 years ago, kind of summarizing what happened in the field of QSAR and the field of machine learning and cheminformatics as applied to molecular representations predominantly and modeling of the data. And this is sort of a summative table that shows that the biggest transformation that has occurred is that we have changed the way we represent molecular structures through machine ready, what we call in the field embedding space representation, and tons of algorithms have been developed, and we have reviewed these algorithms and alluded to the intricacies of specific algorithms and comparison between traditional and new algorithms. And I want to comment on this comparison in the context of the accuracy of the models, not necessarily excitement about new tools, because one aspect I want and I have emphasized and will emphasize in this lecture is rigor and proper metrics that we use, not excitement as a metric, but hardcore data as a metric of model accuracy. And here are a few examination of a few sort of historical papers in the field from the critical, if you will, sarcastic standpoint. And here's one. I intentionally don't include authors because I think that the trend has been and continues, unfortunately, to be common to our field, which is sometimes overhyping, over-describing what actually has been observed. And so here is one of the first deep learning papers using Campbell data. And I highlight the statement made by the authors with the word significant in it, significantly outperform all competing methods. That's a verbal statement. If you look at the actual data and compare the performance of a newly developed, exciting deep learning method contrasted with traditional SVM random forest, that's where the statement originates, that this new method significantly outperform all competing methods. That's if you read it. But if you look at the data, what you find is that the largest performance difference measured in AUC numbers between deep learning and SVM or random forest using the same descriptors was no more than 0.04, keeping in mind the standard error was an order of magnitude larger. And so here is a verbal claim, here is the actual statistics, and what I constantly encourage people is to look at the data and not overhype the results of the modeling, because I think any pharma industry would be hard-pressed to switch from random forest to deep learning if the performance difference is questionable, if at all, and yet it requires greater resources and sort of greater attention to how models are developed. Another story that I find quite symptomatic is when models or model impact is overhyped. And that is, again, very important in the context of remaining rigorous and accurate in the description of what we do. And that's a story about discovering the world most powerful antibiotic that kills every bacteria, every germ, with a lot of publicity associated with the publication in one of the highest cited journals, such as Cell, that was predicated on yet another exciting deep learning method with a new way of representing molecules. And the paper, as you could see as of today, collected over 1,300 citations. So the methodology itself was and continues to be very exciting and very powerful. But looking at the numbers and looking at the numbers in the original publication, the claim was that random forest was significant, and here the significance is much greater than a questionable 4%, 20%, 26%, 40% for different data sets. That's the numbers that were published in this original paper that introduced a new representation learning method and its applications to different benchmarking data sets. That was the original claim. In the original paper, I'm going back one slide, if you look at it today, there is a sort of red exclamation point saying that the article has been corrected, view the notice. If you click on that, you get to the erratum publication. Notice that if the original paper collected over 1,300 citations, this correction was only cited 36 times with really meager altmetric score. And what happened is that the authors admitted that they've made an error in the processing of the random forest model predictions, which is sort of hard to imagine given the complexity of the deep learning method that was developed. But when this correction was made, the claimed advantage of deep learning ensemble over random forest had practically disappeared. As you could see, these numbers, arguably in one case, it was still better model, but in one case, random forest was overperforming by 6%. So I'm showing this example just as a warning that we need to continue to be very careful in how we approach method development, benchmarking, and model validation before using loud words and hype to discuss our studies. And to finish the story that I just described on the methodological part, on the functional part, it turned out that the molecule that was claimed to be this ultra successful broad spectrum antibiotic was actually not new. It was discovered a few years earlier in the same institution, it turned out in the same floor, that speaks about data sharing in academia, volumes. It speaks volumes about this. And it was determined that this molecule was overly toxic. So the previous group stopped its development, but that was unknown to another group that rediscovered this molecule as the world's best antibiotic. So this is just a warning thing. And to finish the warning part of my talk, here is something that I found years ago, and that's a statement from a statistician about science crisis caused by machine learning, and that's my sort of final warning as to how careful we should be with publishing and interpreting models that we develop and discuss and describe in social media. And the observation, which I fully share, has been is that as the data grows and machine learning algorithms consume more and more compute power and process bigger and bigger data sets, it's becoming impossible to reproduce models. Very few people have enough resources to reproduce deep learning models published by very well-equipped groups. And so models are and methods are developed to establish trends, so they always find some trends, and then it's hard to check what these trends are. And so we really, and I will talk about it at the end of my talk, we really should be focusing on experimental validation of hits rather than computational validations, where frequently suffer from data leakage and other problems that people have discussed. And so this is the funny slide, if you will, but there is a psychological rule of repeating yourself multiple times if you want the listener to really learn what you're trying to share. And what I found in the Urban Dictionary, so what happens if we use the phrase AI 3 times? It's a phrase used by most evil people with bad intentions. We don't have bad intentions. We're not evil people. And it's very important for us as we build models to be rigorous in processing data and in interpreting the results. So with that, I will use the remaining time to talk about a few developments, recent developments in our lab, addressing some of the issues that I outlined on the beginning as solvable, interesting and solvable and challenging problems in the field of drug discovery. And the first problem I want to allude to and discuss is the problem of dealing with emerging huge multi-billion compound libraries. And not surprisingly, the need for ultra-fast clustering and ultra-fast virtual screening methods has been articulated in recent literature as one of the acute problems, and continues to be acute as the databases continue to grow. So chemical similarity, the most fundamental, defined and definitive part of cheminformatics, has been okay for a while until libraries had grown. And here is a reasons why chemical similarity search, brute force chemical similarity search, continues to be slow, because it's brute force, because libraries are huge, and most importantly, because traditionally we use high dimensional features such as various types of fingerprints, which, as you know, count thousands of dimensions. And so there are ways of solving these problems, and there are groups focusing on fast clustering algorithms, most notably BitBirch by Raman Miranda in Florida. So I'm not going to talk about clustering, but I'll talk about briefly a methodology we've been developing in the last few years that we've called semantically aware latent space autoencoder, or SALSA, and its little sister called SMALSA, which is small space autoencoder, addressing the issue of how we could accelerate chemical similarity by rationally reducing the size of the chemical space. And so what started this work was an observation by a few students in my lab that when we use standard autoencoders and look at the distribution of embedded molecules in the however sized space of embedding, molecules are distributed not quite randomly, but more sporadically than we might have expected. So that's the distribution of chemically similar molecules in when we use naive autoencoder. And what we have developed is a protocol that we've called semantically or structurally aware autoencoder. And the idea was to build a special purpose encoding protocol that would intentionally place similar molecules into proximity to each other in the latent space. And in order to build this model, we've used series of molecules that were designed to be one graph edit distance apart from each other, and the series of molecules was series were designed in 10 analogs of any given chemical, originally from Kimball. And so using contrastive learning approach, we had forced molecules that were similar to each other in the chemical graph space with universal metric of similarity being graph edit distance to cohabitate proximal areas in the embedded space. And what we had found is that as a result of the special protocol, we have been able to match graph edit distances in the original chemical space by Euclidean distances in the latent space of SALSA initially. And then we formulated a version of SALSA that we called SMALSA that intentionally, so first I'm showing here briefly that our effort had preserved the distance distribution up to 5 nearest neighbors, at least in comparison to a standard autoencoder. So this is the result of SALSA. This is the result of standard naive autoencoder. So you could see that we generally preserve distance between graph-added distance between molecules that we have considered in this exercise. But most importantly and most practical, we have played with the dimensionality of SALSA by progressively embedding molecules in a smaller and smaller and training the model to embed the training set of about 15 million molecules, 1.5 million anchors from Campbell with 10 analogs of each compound. And what we found is that at the dimension of about 20, we preserve the accuracy of embedding while dramatically accelerating the chemical similarity search process. So this is the kind of methods that I think is unavoidable to use as we face the need of rapidly searching multi-billion compound libraries. Another approach that belongs to the same category of rapid similarity searches is an approach that was originated by Arthur Kasov's group over 5 years ago, so-called deep docking, where the protocol of structure-based simulations of docking is replaced by a machine learning model that calculates from chemical structure representations docking scores. So the model is trained on the docking scores obtained for a relatively small number of molecules, about 1 million, initially using standard docking protocols, and then machine learning model is trained to predict docking scores of molecules without actually performing docking. And a series of publications from Art and other scientists who have reproduced this methodology have outlined sort of how this method works and how rapidly it could identify candidates that can then be tested successfully in bioactivity experiments. Not spending overly much time, but my group had published another protocol inspired by deep docking that instead of, this is the core, and I will describe it very briefly because I'm trying to outline the general methodologies and general principles of methodologies rather than specific studies. But the unique approach in our case was the use of generative chemical model in order to produce analogs of high-scoring molecules obtained by original docking, followed by limited docking of these candidate molecules, and using this high-scoring compounds as queries or seeds to identify their analogs in enamine purchasable library, and then repeat the docking exercise. And what we have demonstrated in this cycle of initial docking, generative fine-tune, modeling, and docking validation of selected hits is that we have identified progressively better and better compounds from the enamine screening library, but most importantly, have demonstrated that using these types of methods is substantially faster while producing similar results to deep docking and other methods. And here is a table of looking at the speed. And we could focus and have to focus on accuracy, but where absolutely it's mandatory to use rapid methods. Otherwise, we cannot merely process the huge libraries that are emergent, virtual screening library that are emergent from vendors such as enamine. Of course, and it's very important for any computational methods to rely on experiments to validate calculations. So these are results that we have published as well. Selected use of hidden gem, as we've called the methodology, tool, and software to identify novel inhibitors of a couple of proteins from dengue virus. The last piece that I want to discuss and then conclude my lecture is revising, which is also very important. What we think is important, revising metrics of validation for QSAR-based or machine learning-based virtual screening of ultra large libraries, which is the problem that we're facing today. And the specific acute problem in the field is that most training sets are imbalanced. The virtual screening set is expected to be hugely imbalanced because it's unthinkable to identify a large number of active hits in multi-billion virtual screening libraries, given the specificity of ligand binding to target proteins. And so training sets are imbalanced. Virtual screening samples are hugely imbalanced. And therefore, we need to identify a relatively small number of hits from highly imbalanced libraries. Most importantly, we have to identify the number of hits that can be screened experimentally in medium screening experiments, given the cost of molecules that we need to purchase, even higher cost if we synthesize proposed hits. And so number of proposed hits that or hits that we should propose for virtual screening is usually related to the size of the well plate. And so what we proposed to simulate an elimination task is to use PPV128. So this is the smallest size of the screening plate. Typically it's 384 and compounds are screened in triplicates. So we've used that specifically PPV128 as a gold standard of virtual screening performance. And what we found in the paper titled One Size Doesn't Fit All, published last year, is that for all data sets that we've considered, when we contrast the performance of model optimized to produce balanced accuracy, which is how we traditionally build QSAR models, versus models optimized to produce imbalanced accuracy measured by PPV128 in the training set. PPV in the training set, PPV128 metric in the virtual screening set, we consistently find that models built with imbalanced 1 to 10 or 1 to 20 ratio of active to inactives, we consistently produce much higher PPV128 measured accuracy. So this is something that we recommend doing for virtual screening, that is switch from balanced models emphasizing balanced accuracy to models emphasizing imbalanced accuracy and positive predictive value as a chief metric for the training set models to be used properly. So here's the concluding part of my lecture, and I will introduce a new line of research that I find very inspirational given where we are in the field. And I'm coming back to the slide that I showed before, saying that in the beginning of the talk, saying that we seem to have very large chemical bioactivity data, and our challenge is to find molecules in the virtual screening libraries. But the data sets that we currently have are really not that great. Here is a sort of summary figure that Lishbai had published on the website, and that's a very interesting, philosophically interesting observation that historically the models and methods that have been head-turning in the last decade featured the same observation, and that is that the methods that have been used to fuel Atari, object classification, Jeopardy, and more recently, text generation and protein structure, methods have been developed in advance of accumulating enough data to make the methods work. And when we put together methods and data, what emerges or emerged in the last 5 years are monumental accomplishments such as text generation, accurate text generation, and protein structure prediction, both earning high impact and praise from the entire society. And what we perhaps underestimate is, and the value is that these developments have been enabled by a combination of rigorous algorithms and really enormous size, high quality data sets. These are the number of data, the amount of data in hundreds of billions that has been used in order to train these models. We don't have this type of data yet in chemical biology. But what I want to talk about is that this data is emergent, and I encourage you to read a paper published by the Structural Genomics Consortium that talks about the challenge of generating enormous amount of modern chemical bioactivity screening data to fuel drug discovery and enable the field to transition to much more accurate models and much more impactful models. And so one of the areas that experimental areas that fuels the transition is the technology that has been progressed in the last couple of decades, DNA encoded library. I'll be very brief. What has been demonstrated by one of the companies, pioneers in the field, HitGen, is that the models built on DEL data can be used for virtual screening chemical libraries. So another example of virtual screening, such as to identify analogs of DELs and replace off-DNA synthesis, which is costly and time-consuming, by screening of purchasable compounds, so sort of standard historical cheminformatics approach applied on top of DEL models, with a caveat that DEL libraries contain billions of molecules. So we're really talking about a new wave of QSAR model development. We published a paper last year as one of the first exercises in the open science domain, processing a few proteins and demonstrating that by processing about 3 billion size data set with about 20 to 30,000 compounds labeled as actives, we could build QSAR models and in one case achieve an elimination of 50 compounds, of which 7 were found to be active. Going briefly back to the property prediction, we've used multiple property filters, including software that's called Stoplight that eliminates poorly designed molecules and poor physical property molecules, and found that in one case we were successful. We were successful only in one case, as we rationalized in this paper. And I need to finish in a couple of minutes. Kushal, I see that, Kunal, I see that you want to stop me. I have a couple of slides left. But what we found going to the basics of QSAR modeling is that the reason why our success was limited is that the chemical space, and that's a very well-known problem in machine learning and cheminformatics, the chemical space of DEL libraries in yellow was non-overlapping with the chemical space of the virtual screening library from NMIN. So this is a challenge for designing focused DEL libraries we're addressing today that produce models that can extrapolate better and generalize better toward NMIN. I'll skip that. So what I want to say in the end of the talk is that the data that enables new wave of machine learning and new combination of big data and algorithms are emerging as a result, especially of the efforts by the Structural Genomics Consortium with the recently funded Ligand AI Target 2035 project, which is established to produce billions of chemical bioactivity data points. So this is Ligand AI website. I encourage you to visit, with the database built by my colleague Benjamin Hype Keynes in his lab at the University of Toronto called ERICheck, which stores or will store all this data on DEL screening. And there is a symposium in about a month and a half in Barcelona, so-called Mainframe, that Albert Antolin is organizing, where people for the first time will get exposure to this new data sets and will start new challenges to build more models of previously unseen chemical bioactivity data. What we are facing is a challenge of accelerating drug discovery, and this is a paper that recently summarized the trend of the field and where CAT can dramatically accelerate, especially preclinical drug discovery pipelines, and that's where DEL projects fit the best. So this is the summary of the talk. This is the challenges that we face. Model increasingly large and complex databases and ultra-large chemical libraries, high efficiency and prediction accuracy, something I did not talk about, but models that replace animal testing, models that integrate with machinery to produce self-driving labs, and ultimately achieving clinical impact. With that, thank you very much. And the last statement I want to make is how we should achieve discovery in our field by combining modeling and curated content, which is the new interpretation of the famous formula. Thank you very much.
Thank you, Alex, for this nice presentation. I'll quickly take one question from the chat. Prof asks, as of now, there has been limited exploration on the identification of activity clips that can potentially mislead the QSAR. Can you comment on the importance of their detection and the cheminformatics methods proposed so far?
That's a great question. And of course, as you know, there have been multiple attempts to solve the problem. I think the problem continues to be unsolved. The biggest question is in the data sets that we process, how common is the problem of activity clips? If it's overwhelming, the models cannot be built. And we have published an approach that we called modelability assessment that effectively pre-identifies the number of activity clips in a given data set. If a data set that you model is overpopulated by activity clips, I don't think rationalization is possible, to be honest, and those representations then need to be addressed, and perhaps different methods need to be used. But that is certainly an important problem.
Yeah, and can I ask one question quickly? So what is your opinion on the increasing use of descriptor-free language modeling in cheminformatics?
I think it's so we discussed this, Kunal, in the deep QSAR paper. I don't expect particular miracles to come from descriptor-free models. But I think the advantage that we have is that we can engineer this. They're still descriptors, right? They just the representation is engineered rather than calculated using predefined formulas. And so we could improve some of the learning tasks. The SMALSA method that I discussed is an example of work in this direction. And I think for the tasks such as chemical similarity searching, I think these types of methods are critical because they offer acceleration while preserving accuracy. I am not sure about QSAR. We're investigating this as we speak, essentially, as to how to build lower dimensional models with high interpretability. But I think it's a very interesting and important area of development. I know that you work a lot in this field recently, and that's the kind of work I think that should continue with proper benchmarking.
Thank you very much, Alex, for your time and nice presentation.
Sure, I'll stop sharing. Thank you.
So our next speaker is Corina Martinez-Mayorga, who is a researcher in cheminformatics and computational chemical biology at the Institute of Chemistry, National Autonomous University of Mexico. Her research focuses on predictive modeling of bioactivity, representation, and descriptor selection, with particular attention to the limitation of QSAR and machine learning models in complex biological systems. She has published extensively in leading journals in the field, and currently she serves as editor-in-chief of Journal of Cheminformatics. Her today's talk will be on rethinking of bioactivity modeling, representation, context, and predictive space. Corina, you have already shared your presentation. Please start. You can start.
Thank you. Thanks so much for the nice introduction. And thank the organizer, Tudor Oprea, Conan Roy, and Abdurham Olat for the kind invitation to participate in this online seminar series. And I'm also very honored to share this space with Alex. And after this wonderful talk, you cannot help yourself but take some notes and learn. So I took the liberty to take some screenshots of your talk, Alex, and I put it in I just visited my talk, so I might be pointing out to some of the things that you were discussing. So it was a really, really inspiring and very relevant presentation, I would say. So you can see my full screen now, right?
Yes, it is visible.
Perfect. So for those that are not aware of where we are working, we are located right here in this place right here. We are like 4 hours drive from Cancun, and it is a very, very nice landscape. This is called Yucatan, in the Yucatan Peninsula, and these are some actual pictures from the area, and it's really, really beautiful. It's not like AI-made figures. So I encourage you to come anytime you are on this side of the world. So thanks everybody for attending this presentation, and this is the outline of my talk. So I'm going to start by saying some things that I'm not be discussing, unfortunately, then the motivation of this talk, and it is going to be divided into 3 main parts. Like I'm going to be mentioning some points on representation, context, and predictive space. I will be spending more time in the context part. Then, rather than concluding, I'm going to open questions for the community and present some take-home messages. So unfortunately, in this presentation, I don't have the time for discussing the work that all the people in the lab has been doing. But still, I wanted to show this picture of the current members of the group and some also undergrads, grad students, and colleagues. So, and is the list of people that has been in the lab previously or nowadays. So we work in the interface of cheminformatics, bioactivity modeling, and data-driven analysis with systems that have to do with chemical or biological intentions. So the ideas that Alex presented are really, really important. And so here are some of the snapshots that I took and some things that I want to point out. So Alex was mentioning something really, really important. That is, how are we evolving on this field of QSAR and predictive modeling as a whole? So there are different ways and different places where we can improve the modeling that we are producing. And as he was saying, there is a lot of hype or a lot of work doing on different areas like developing more algorithms or things like that. But maybe it's time to think on different aspects that are also hidden within the data that we have. So the ideas that I'm going to be presenting start like in these publications that are shown right here, where there is some interesting papers, and actually some papers of this list are from the Tropsha's group that describes some of the 21 errors encountered in developing QSAR models. And interestingly, if you check these errors are still happening. Some of them are avoidable and some are really, really hard to avoid. It is interesting to see how many of these avoidable errors are still happening. So we should be paying more attention into these because it's something that we have in hand for modifying and editing our pipelines for improving our models. Some of the things that I want to emphasize is that we need to bear in mind that we are oversimplifying how we look at the systems, how we look at the data. And I'm going to talk about that in a little more detail. So we are doing some oversimplifications and assumptions that are going to limit the precision of our models. So Tropsha already mentioned a lot on increasing the data size of our databases, and which is actually really important. And also we could think on certain improvements on the algorithms, although that might not be kind of so much striking, but still we can have some improvements there. But what I'm mentioning here is that we need to take into account the context of what we are exploring. And this is actually the key part of this presentation. So the boundaries on the predictive models might not be particularly technical. It might be how we are looking into the data. So for analyzing that, I'm going to be talking about what I said, as I said, representation context and how this affects or let us build the predictive space. So for those that are very familiar in the topic, of course, we have our basis in the representation. But when we decide to explore an endpoint, when we define the endpoint, we are already introducing some simplifications and that may obscure complex mechanisms. So if we are interested on any particular endpoint, we might be ignoring other effects that might be happening and are happening. So the representation that we select will adapt to simplified tasks, but will not give us the entire picture. So for that, what we are representing actually determines what we can learn. So we need to be careful on how are we defining the endpoint. And definitely, I agree that the good data and validated models are mandatory, but we need to think that the validity of that data and models are context dependent. And I hope at the end of this talk, we will kind of clarify what I mean by context. So for the people that is starting on this field, I just want to mention how we use the descriptors as a language, of course. So descriptors are used to translate the molecular structure in numbers that we use to develop our models. These descriptors, of course, ideally will emphasize on different properties of the molecules and will make some parts of the information will become explicit, but some will remain like hidden or invisible. And that's the tricky part. So there are different ways to analyze these descriptors. And as Kunal was mentioning, we can even have like a descriptor without descriptors. But I'm pointing to another direction here. So we can have other types of descriptors that are not very much used yet, I would say, like the quantum-derived descriptors. This will give us a complementary view of the molecules, which is something interesting. Of course, they will give us a view with emphasis on the electronic side, like the density-based features will become explicit. And of course, this will provide us a different chemical space. So we will have a different way to explore how do we compare these molecules. But of course, I agree that we need to think if we really need to spend time and effort developing quantum chemical calculations and introduce that in our model. Do we really need to care about electronic density or not? For one of the methodologies that I really encourage you to take a look is the QTIM method. It is based on the atoms in molecules theory developed by Vader in Canada some years ago. And it's actually quite an interesting method. So in this slide is courtesy of David Ramirez. And he is a very much expert on this method. He was doing a postdoc in a group, and he is able to produce this type of analysis. Using this atoms in molecules theory, you can develop to have the molecule using quantum chemical calculations, of course, and you can describe the atoms and the bonds based on physical terms. It is not like you just set a threshold of distance and you assign a bond. You extracted that from the density function in the calculation. And something that is really, really interesting is that what Adrian Royberg has been developed and is called ANI. For those that do not know ANI, I encourage you to look at this area. And this is a machine learning-based model that gives you some precision that is really close to a very, very high level of theory in quantum chemistry. with a very, very short time. So it's very precise and very fast. This is an example for for this type of data. Using DFT, it took like 52 hours, and using ANI, it took 0.3 seconds. So Alex was mentioning how if you have a difference in precision, like a very, very, very small, is not worth. However, here we can see that it is extremely fast. And is giving a very good results. So what I mean here is that now we have access to information that has a quantum chemistry level of data with a very cheap, computationally cheap calculation. Of course, it has some issues, but I think it's still worth checking. So in these presentations, there will be some parts of the information that will not be explained, and that part is what will raise error. So these errors are very important, for example, when we have like a multi-target processes. And of course, we should expect see more errors if we are dealing with a multi-target systems. And this is something that is kind of intuitive if we look into the biology side. In some cases, if we use nonlinear models for describing these systems, it can explain a little bit the discontinuous SAR, at least partially. So one of the studies that I found interesting was this developed some years ago by Jürgen Bajorat. In this study, they were analyzing the activity cliffs to describe how we can understand these activity cliffs. So if you understand what's going on, you might be able to at least adjust your ideas of how to develop your models. And what they did in this study, they took some molecules, similar molecules in the PDB, like co-crystallized with some proteins, and they analyzed the SAR. And sure enough, they were able to give some explanations for the activity cliffs based on the interaction with the protein. However, not was sufficient. In some cases, it was not possible to explain the full problem. So what happened there was some information that was missing. So, and this leads me to another idea that we encounter. So, and I'm going to come back to that I said with Jürgen Bajorat. So, also some years ago, we were analyzing this protein. This is a prohormone convertase protein that we were having this collaboration with Iris Lindberg in University of Maryland, and then this protein has relevance in diabetes. So we did like the screening, and we were very, very happy because we had like a very nice computational lead. And it turns out that at low doses, this molecule stimulates the response. But if you increase the dose, it inhibits the response. So you have like a different activity in the system. It is not like a potency. It is like shifting from stimulation to inhibition. And the way that we explain is that you can have like different binding sites where this molecule can bind. And this means that you first saturate one site, say the allosteric site, and it exhibits the stimulated response, and then you start getting the molecule into the other side, and then you have the inhibitory side. So you can have like different effects, and we know that from the biology of this type of proteins. Maybe that's the reason why in analyzing only one binding site is not enough to understand what's going on in the interaction of these small molecules with these proteins. So this tells us that the dose is important for saying where are we going to like direct our study. So this is what we are calling here like the context. So the context will be the, for example, the dose that we are using. So we have the representation for the molecules, but then we need to take into account the context in order to define very well the endpoint that we are going to use. So relevant predictive limitation will be introduced before the modeling begins, when we are defining the conditions of the experiment. So in bioactivity prediction, we are using, like in the root, the SAR, the structure activity relationships. So we are focusing on the structures, but as I said, we need to take into account for example, those to derive what structures we are analyzing. So, of course, the QSAR machine learning models in general are formulated in terms of structure activity landscapes, and smoothness is assumed or hoped for, but is, of course, not guaranteed, which is the case of structural activity cliffs. What I think is going on is that we are implicitly assuming a fixed experimental context. So we are assuming or hoping that everything is going to work on that scenario. Again, context matters, and this could be kind of a blind spot. So if we do not pay attention into this, we might have some errors. So molecules can have like can behave different under different contexts, as I was mentioning in the performance convertases case. Ignoring this context might collapse the different biological scenarios that we are using into one single point, or like a hidden group of activities inside the same analysis. Some of these factors that affect the assay, some of these contextual factors are, of course, dose, the asset type, the biology system, temperature, time, etc. And it was interesting because this also came as a discussion with one of the colleagues that we have here in Mexico. We were discussing, and she started to tell me the experiment that she was running and the problem she was running into. It was really in line with what I'm describing here. So this led us to understand a little more on what was going on. So the context is not, I think we can say that the context is not noise, but is part of the signal that we are explaining, that we are studying. So the predicted models will behave well of course, inside the predictive space, in the applicability domain that we define. But we cannot switch from one predictive space to another, because they are defined with different predicting variables, for example. But the molecule doesn't care. The molecule can bind to different places and will be switching from one endpoint to the other inside the same molecule, inside the same assay. So again, the predictive models can be stable and well-behaved within the same well-defined predictive space. Changes in the endpoint of definition or experimental context will induce different predictive spaces. If we, by any chance, mix those endpoints by ignoring the experimental factors, for example, we will be merging chemical spaces and then predictive spaces, and this will not be very effective. And then the apparent failure of the models might reflect these transitions between different predictive spaces that are due to contextual or mechanistic shifts, not necessarily to technical flaws. Again, experimental context will becoming crucial. So all these ideas are described in this paper, in this publication that we call Disruptions in Bioactivity Driven by Dose, a Challenge for Drug Discovery. And so when we submit this paper to the journal, I was skeptical because I wasn't sure if the community was kind of understanding what we were saying here. But luckily, the reviewers were very supportive, and knew that it was something worth mentioning. So these productivity disruptions are intended as how we are modifying the activity, not you can think of it as an activity cliff, but not because of the change in the structure, but change in the context. And you can see more in this publication. So then when we were analyzing this paper, we wonder if there was another cases of activity data that described those dependence. And sure enough, we found many examples in the literature on many different levels. So the dose is really changing how we are looking into the data. So we can have a different activity by just changing the dose. So this is only one of the contextual variables that I was mentioning. There are many, many others. So, but this, I think, shows that there are many examples in the literature, in the experimental side, that is telling us that to pay attention on how this is behaving. So, if you look into the pharmacological literature, you can find different ways that people describe this information, and they are actually like different models that they use to describe these trends. And this is also described in the same paper. So let me put this in this slide, kind of the summary of these steps. So we were moving from a conceptual questions to a modeling framework. So what the conceptual observation was that we've observed that the same molecule produce different outcomes, suggesting that the context, that the coexistence of the distinct endpoints. This is what I mentioned about the pro-hormone convertases. Then these points these point us to a contextual effect other than just modifying the structure. So we hypothesize that the apparent failures in modeling may arise from implicit mixing endpoints rather than the model limitations. So then we took the task of looking into the literature, and yes, we found a variety of examples consistent with the endpoint-dependent behavior. So these effects are not are often dose-dependent, but there are other contexts that affect this dependency. And such transitions from one activity to another, from one endpoint to another, may occur and actually happen for the same molecule, leading, of course, to this bioactivity disruption, which is actually the title of that paper, precisely. So then we wonder, how can we understand further these ideas? And if you look into statistics, you can find that these effects are captured by what the statisticians call contextual covariance. So we could incorporate this information into the models using this contextual in the framework of contextual covariance. So, but here we are framing this as an element for defining actually the chemical space that we will be analyzing. And so now that I mentioned the chemical space and the predictive space, I think it's worth mentioning like the difference between these two. For some people in this audience, of course, this is very quite obvious, but I think it's worth mentioning for the general audience. So, of course, they are not equivalent. The chemical space is meant for organizing molecules based on general uninformed representations, and the predictive space supports inferences about a specific property. So the chemical space in general serves a different purpose, like the example that Alex was mentioning of comparing different databases, but for doing analysis, comparisons, similarity searching, etc., what we should be using is the predictive space that, of course, will have like a much less features. And this is, you can argue that this is just for traditional machine learning models, and that's correct. but still in deep learning area, this is done inside the model. So I think this sentence here summarizes what I was pointing out. So the predictive space is the endpoint dependent, and the endpoint is going to be context dependent. If you change the dose, for example, or if you add cofactors, or if you modify the context variable, you might be changing the endpoint, and therefore the predictive space must be redefined. In this situation, the predictive space is then endpoint dependent and therefore context defined. So now we are moving from the representation of the molecules to how to make inferences on the model. So of course, the representation will define what information is available. The context will define the endpoint. And the predictive space is built or is constructed from the interaction of these two, the context and representation. So the context will define the endpoint and, of course, the predictive space. So I think this figure kind of summarizes what I said and is kind of in a visual way. In the top part, let's say that we have this chemical space represented here by these Lego pieces. And, I should have here, this same figure should be right here. And then we have like the endpoint A and the endpoint T. If everything works perfect and very clean, we can start from the chemical space and develop the predictive model using the endpoint, selecting the features and building our model, in this case, a tree or an animal, for example. But what happens if we have like these two endpoints mixed together, like in the case of pre-hormone convertases, just like an example? So if that happens, we will be getting the descriptors from the pool that describes actually both scenarios. We will need a context variable to separate endpoint A to endpoint B, and then we can build separated predictive spaces for each case. If we do not do that, then we will have like a mix of a model with a lot of error. So the context, like here, will define the endpoint, and with that, we will construct the predictive space. So what could follow after this? So a practical next step could be the explicit endpoint documentation, the context stratification before modeling, multi-level modeling, like in a hierarchical way, and making those aware modeling pipelines. And these are also some screenshots that I took from Alex's presentation, which is in line with what he was mentioning, that we really need to get like quality data. And by quality data, we mean not only like the good measurements of the experiments, but really paying attention on the context or the different variables that are used in the experiments. So this is kind of what I wanted to mention to you. And then in the next few minutes and few slides, I want to raise some issues that I think are relevant to think of. Instead of conclusions, I will delineate some emerging issues. So many of the challenges persist despite the technical advances, which is in line with what Alex was saying, and we need maybe to pay attention in conceptual and semantic issues. And some problems will require reflection in addition to optimization. This is one of the ideas that I think we could have in mind, is if the existence of property cliffs. Do property cliffs exist? If you think about it, they shouldn't exist, right? Because that's the basis of additivity and transferability in chemistry. But they do exist, but they are very, very rare. why they are rare? Because their properties behave as a continuous additive functions of structures governed by deterministic incremental contributions of structural features. And importantly, they are often chemically interpretable, so we can understand what's going on around those property cliffs. In turn, if we think in terms of the biological side, the activity cliffs, like the structural activity cliffs, that is the common name that we use, are relatively frequent. We don't know exactly how prevalent they are, but they are more frequent than property cliffs. They are often difficult to rationalize because they emerge from complex, nonlinear, and threshold-sensitive processes. And of course, they can have a strong impact on predictivity. So it's important to remove those cases because they cannot be used in the models. And this activity cliffs is kind of the similar idea of the disruption in bioactivity. But in this case, that's why I'm emphasizing that we should call it like a structural activity cliffs, because they are based in the definition of activity cliffs of the structures. But the breakdown of activity could be happening for the same molecule. So that's the disruption in activity. And not all the cliffs have the same epistemic status. This is a courtesy of Elian Abreu. So then moving forward, I suggest to question a little bit what we are doing in the SAR paradigm. I already questioned the S part, like the structural side, saying that we can have like differences in activity based on the dose, for example, and many, many other contextual variables. And we can also argue that we can rethink if we really need the R, or if we can use another term or idea. What happens is that some of the analyses that we do are not actually relationships, but in mathematics can be more close to associations, which is quite different. And then, for instance, this figure shows really clear that for simpler systems, we are very close to the structure-activity relationships, but as we move away and we use more complex data, we will be analyzing more associations and the differences in the activity based on the structure might not be relevant. That might involve other aspects as the context. And of course, we can have properties or activities. This idea is summarizing these publications this year and is in close collaboration with Jose Medina, which is the lead author on this publication. And we put together that paper. Another aspect that I want to raise here is the idea of how many terms we are introducing in the literature, like the X-informatics here, for example, where everything that we are analyzing, we put this informatics part. However, we are having a proliferation of X-informatics labels, and we are maybe addressing similar problems on the different names. I totally agree that we will have differences between the fields, but we have common things as well. At some point, I was checking some information of matrices that we use in QSAR, and I was shocked that I found exactly the same equation in another field, and they have like a different name and a different numbers in labels in the equation. It was actually the same. So maybe we can look in a little bit in different areas, and we can have ideas or even things that we might borrow. So probably, and this is like an open idea, we can build a comparative typology that reveals shared epistemic and infrastructural patterns across established and emerging informatics domains. And why not? We can propose a criteria and guidelines to assess the legitimacy and maturity for proposing a new informatics field. So some of these topics are very mature, and some are not. And if we look at this figure, for example, the bioinformatics area is quite like very mature, and probably this is the reason why many people still uses bioinformatics and cheminformatics interchangeable, which is not actually true. Well, good, but of course we have some common analysis that we share. So my point here is if we are gaining clarity or we are just adding more labels. Again, this is a courtesy of Elier. And then another topic that I think is worth mentioning is how we are using this term of AI. And this is something that Alex also mentioned in his talk. And I'm just going to raise here this idea of using the proper language, because I've seen some papers saying like AI-driven, AI-based, AI-supported, and they are used interchangeably. If we are using AI tools, doesn't mean that we are designing or developing AI itself. An example that came to my mind is like if you are using a force field parameterized with quantum chemistry, doesn't mean that you are doing quantum chemistry. You are using the information out of it. So I think we should be careful moving forward in the precision of terminology. So with this, I want to say my take-home message. That is, the context is not noise, but because it defines the problem. And here the figure is complete, by the way. So apparent failures may arise from how the molecules are represented, how the endpoints are defined, at what models we are asked to explain. So in the next slides, I'm going to share with you the news and the idea of publishing in the Journal of Cheminformatics. As Alex mentioned, I'm now the editor-in-chief, along with Barbara Zratsil, that works in Campbell. And this is a wonderful group of people that we are behind this journal at the moment. These are the associate editors. We are just missing here Jane. This photo was taken yesterday in our monthly meeting, and I encourage you to participate in the journal. Like you can approach with ideas or things that you want to know. This summarizes why, what, and how to publish in the Journal of Cheminformatics. This is taken, of course, from the Guide of Authors, but you can approach us for different ideas, for example, like to review papers, to publish, to participate as a guest editor, and we are very much open for your ideas. I also want to share that we will be holding the School of Cheminformatics in Latin America. This has been done 2 times every 2 years. And this is the people that participated, I think, in the first one. And the second one was in 2024. So we should have it this year by the end of the year. And so stay tuned. This is done in virtual format. So I'm sure we had people from India as well. And I know in this audience we have several people from India, and that's very nice. So the online version helped us to have this broad audience. And I'm going to finish this presentation with the keywords that build the research that we are doing, and one keyword that I think we need around the world these days. So to finalize without something artificial and more human, I want to share with you some of my drawings and some of my hobbies. So I hope you enjoyed this talk, and I'm happy for taking any question.
Thank you, Corina, for this thought-provoking lecture. Before I go to the question in the chat, I will ask you one question. So you have mentioned already about the dose dependence and context dependence predictive space. So do you think that this applies more for the classification-based modeling, because in case of regression-based modeling, we use mainly say IC50 or EC50 like endpoints, which are derived from multiple dose measurement, but finally we convert them. We get the values in terms of EC50 or IC50. That is actually the dose required to elicit a particular fixed response. So here that in that case the response is fixed. We are taking only the dose required to get that response. And also, as per OECD principle 1, we have to use the same experimental protocol for developing a regression-based model. So we usually do not mix data from multiple context-dependent measurements in the same regression-based model. But in case of classification, we do so. The data may come from different experiments with different context and different conditions, where defining two classes, maybe active or inactive, or toxic, non-toxic, becomes the context dependence, and there your proposition becomes more valid. So do you think so that your context dependence predictive space applies more in case of classification-based modeling?
Definitely, I think yes. That's a very good point. The first step could be like a classification model, just to say like if it is going to go for one, let's say one endpoint or, instead of the other, and then after that you can develop a predictive like regression model. Yeah, that's a good point. Yeah, although the other part might also happen, right? But yeah, that like this could be also seen as a classification model before the regression model, definitely.
Yeah. So now questions from the chat. The first one is from Marco Provo. Similarity can be defined by two different ways. First, similarity among compounds using selected feature space obtained using supervised algorithm. And the second one is considering the entire chemical space. For example, some fingerprint or using Tanimoto similarity or Hamming distance, which is unsupervised. Do you think that visually very similar molecules may not always generate efficient predictions, while a part of visually not so similar compounds may generate improved predictions due to supervised aspect present in the selected features?
Well, if I follow correctly, the question is if visually we can have like an overlap of like a close two molecules that are close related or in the same space, and if they might not have the same activity. So the visual part is something that we need to be careful. Sometimes we should not rely so much on the visual side. For example, in a chemical space, for example, if the variance is not well explained. If the variance is well explained and if the properties are defined for the activity, yes, the activity of two neighbors should be similar. That's the principle of similarity, but still we have the option of these activity cliffs. So the answer is yes, but just in the case that you are selecting the properties, the features that matters, and the visual, the graph that you are building gives you a very good explanation of the variance. If you do not have these two things, then the visual might not be very helpful, and you will have very likely two molecules that seems different in the space but are very close in activity. But it is not activity cliff. It is just like you are not adding the proper descriptors in the graph.
Yeah. Okay. The next question is from Eric. To represent the chemical space, we use descriptors such as fingerprints or physiochemical descriptors, etc. But which are the descriptors of the predictive space that you are talking?
Well, I was focusing more in the physical-chemical descriptors, but the predictor space can be built with any representation that you are using. It is in mathematics is the space where the molecules live and have this distinction in the activity, in the way that you want to build that space.
Okay, so I don't see any other question from the audience. So in that case, I thank you very much for your time and nice presentation.
Thanks so much,
Corina.
Conal. Good to see you.
So with this, we come to an end to the present online session. We hope to meet you again in the next online session, tentatively in May 2026, and that will be hosted by Abdul Rahman. We also hope you to see in EuroQSAR 2026 this September, which will be held in Italy. So you may please visit the qsar.org site for more details and enjoy the rest of your day. Goodbye.